I'm Sophia Ryan. My intern was Emma Fox. Emma just about to begin, I believe, 4th year in the University of Texas at Austin. Her aspiration to become a researcher, a scientist. She's particularly interested in virology and viruses in general, including viruses that may cause cancer. She actually studied in my lab something, some shared property between some cells that infiltrate tumors and viruses. And this is the ability of cells to fuse with other cells, with the genetic capacity. So that's something that happens in tumors, and it also happens with viruses. This is how viruses actually enter the cell. A very ancient mechanism, very interesting one, not well understood, not well studied. And we actually were able, with help of Emma, to discover a couple of really interesting new things, and she's going to tell you all about. Just to give a little bit of background on what she's going to talk about, we study breast cancer, and specifically breast cancer spread. And the spread of any tumor typically occurs through blood vessels or lymphatic vessels. We specifically look on lymphatic vessels. It's very common mechanism of spread in breast cancer. And in order for these lymphatic vessels to form, this is preceded by infiltration of tumor, but sometimes they're coming from the bone marrow-derived progenitors for lymphatic vessels. So what we discovered recently in the lab, that these progenitors have the ability to fuse, and this is how they deliver the messages from bone marrow to lymphatic vessels to continue to increase their density. And when the density of these vessels in tumors increase, so increase the metastasis. So this is why it's so important to understand that, because the idea is that if we can intervene and interfere with this process of spreading in very early stages in breast cancer, we may be able to at least reduce, if not eliminate, metastasis, which is, as we unfortunately well know, as a major cause of mortality from cancer. It was absolute joy to have Emma in the lab. I selected her application. I looked at this application and I said, I want this job. I want her in my lab. I want to train her. And that was wonderful summer. We had a lot of fun. I think that's all what I want to say. And Emma will present the project.
Okay. Hi. So tonight we're going to be talking about the fusogenic profile of myeloid lymphatic progenitors recruited to human breast cancer xenografts. So the main idea of this, as Dr. Rand was just talking about, is lymphadogenesis, which is the creation of new lymphatic vessels. Now, lymphadogenesis, although it occurs lots of times during embryogenesis and as you're developing, your lymphatic system is growing with you. However, once you're an adult, it mainly only occurs in chronically inflamed tissue, which includes tumors as well as wound areas. And it can be induced by many things, but as Dr. Rand was just talking about, bone marrow lymphatic progenitors are a main cause. And the term bone marrow lymphatic progenitor just refers to a cell that comes from the bone marrow, is lymphatic, a part of the lymphatic system. It is a progenitor, so it has not yet differentiated or specialized into another cell. And the main reason why we're studying lymphadogenesis, as Dr. Rand was just saying, is that as you increase the amount of lymphatic vessels, you increase the rate of metastasis. Now, lymphadogenesis is not causing metastasis, but because lymphadogenesis, you now have more routes of dissemination for the tumor to spread, metastasis can increase once the tumor decides to spread because you have more roads to pass through. So again, the metastatic process, you start off with the tumor, and from the tumor through the lymphatic system, it goes to the sentinel lymph nodes, which are throughout your body. And from the sentinel lymph nodes, it travels to the regional lymph nodes, which are the ones more people are familiar with behind your neck, in your groin, behind your knees, and so forth. And from the regional lymph nodes, then it spreads to the organs. And this is known as distant metastasis. And as Dr. Rand was just saying, this is why we study what we study, because distant metastasis is the main cause of patient death. So if we can target distant metastasis, we can hopefully prevent lots of mortality. So again, this is the conventional route of metastasis, as is understood by many at the moment, and is that, as you see on the far left, you have your gray tumor cells, and because the body can view the tumor as an organ, so it wants to feed it, wants to provide it nutrients. So in come the blood vessels, in come the lymphatic system, and they start to feed it and bring in food. And then in comes the immune system, sometimes to help, sometimes to hinder. So you have your stromal cells, you have your tumor-associated macrophages, and you have the tumor themselves, and they supposedly are secreting this growth factor, known as vascular endothelial growth factor C, that is then taken in by the lymphatic system, and then processed, and then it causes the lymphatic system, as you can see in the fourth image, to sprout. Now you have these all new routes of dissemination for the tumor to see, as you can spread. However, in Dr. Rand's lab, we have not found any evidence that VEGF-C is being produced by these tumor-associated macrophages, which is why we are suggesting that there is another pathway, an alternative pathway, that has to do with, instead of a soluble factor being produced by the tumor itself, it's an innate property of the cell. And these cells we're going to be talking about are going to be referring to them as MLACPs, which is very fast. But it is myeloid lymphatic endothelial cell progenitors. Myeloid meaning they're bone marrow derived, lymphatic, they have lymphatic markers, endothelial meaning they're on the surface, and they're again a progenitor, so they're not yet differentiated. And we know these are a high concentration of breast cancer patients. We see it both in biopsies and in blood, especially when compared to healthy patients. And again, it's already known from existing literature that they expand the lymphatic network, expand your lymphatic vessels. And what is very special about these MLACPs is instead of being just myeloid or just lymphatic, they are both. So they have myeloid markers such as CD11B and lymphatic markers such as LIVE1, which we actually looked into a lot. So this then creates the question, how do these MLACPs expand the tumor lymphatic vasculature? We know they're doing it through cell fusion. So cell fusion is when two or more cells fuse together and fuse their genetic information. So it's already known to be involved in the most famous case of embryogenesis when the sperm and the egg form the zygote, placenta formation, osteoclast formation, multinucleated giant cell formation, and muscle cell formation. There's also evidence for cell fusion being involved in cancer formation and metastasis, as well as angiogenesis, which is the creation of new blood vessels. However, the exact mechanism of this has not yet been fully ironed out, so people are still researching that. So the way fusion works, as you can see here, in the top left image, you can see that cells are always recognizing each other, right? They're releasing cargo, and they're always receiving cargo. So if the two cells like what they're seeing, then they'll begin to approach each other, and then the outer layer of the phospholipid membrane will fuse together in hemifusion, and then the inner membrane will fuse with pore expansion. So now you have a cell that has two different nuclei, and again, these nuclei carry genetic information, so you have a cell that has two different types of genetic information. This is known as a heterokaryote, and then sometimes the two nuclei fuse together, which again, not completely understood at the moment, and then to one nucleus. So this is showing that cells can transfer their genetic information in varying ways that is not reproductive. And this is important because cell fusion has been shown to happen with bone marrow progenitors in regeneration. So again, bone marrow progenitors, but not MLACPs at the moment. So then this begs the question, are these new lymphatic vessels also formed by fusion? Which is why we are hypothesizing that these MLACPs that are recruited to breast tumors might have the potential to fuse with pre-existing lymphatic endothelial cells, thus reprogramming them as more proliferative state because they're exchanging their genetic information, and again, as they're more proliferative, they can then cause or lead to more metastasis, which unfortunately leads to more patients' death. The way we approached this was by determining the expression of fusogenic markers in the tumor MLACPs. So we did this via immunohistochemistry, as John was talking about, and we did a double staining of human breast cancer xenografts for known fusion regulators, again, through past literature, and the lymphatic marker LIVE1, which we already know, as Eva said, identifies those MLACPs. So a human breast cancer xenograft is a new term for probably most of you, and this refers to a human tumor that is then taken and put into a mouse model. So the tumor cells themselves are human, but everything around, so the microenvironment, all the cells that are being recruited, are mouse cells. However, the mouse model is used often in cancer, so what happens in the mouse generally also happens in the human, which is, and we cannot use human studies for this for ethical reasons. So if we look at immunohistochemistry, you can see there on the right, this is what we did basically all summer. So you have your fixed tissue, let's say it's a breast cancer sample, and then there's a bunch of antigens on it. And antigens are just proteins, amino acids, etc., that you can bind. And antigens do not have to be a bad thing. And then a primary antibody. So let's say our antigen is LIVE1. We're looking for that lymphatic marker. We introduce a primary antibody that is complementary to that antigen, so to LIVE1, and then it will bind. And then instead of having a fluorescent probe on that, because that would produce a weak signal, we then introduce a secondary antibody that is complementary to the primary antibody. And this secondary antibody does have a fluorescent probe on it because it can bind the primary antibody at multiple points, as you can see here in orange, so that it creates a strong signal on the microscope, like you can see here. So what we are looking at in the green channel, that is the lymphatic marker. And then in the middle red channel, that is the fusion marker. And on the far right, you can see yellow. So in the green and the red mixed in yellow. And you can see that every single one of those live blood cells, those lymphatic cells, are expressing fusion markers. And this is a different one. So this is, again, that same lymphatic marker looking for MLECPs. And then in the middle, it's a different fusion marker. But again, in the far right, you see complete overlap between those live blood cells and those SIRT-beta-1 cells. And our final example of the night, again, representative images. We see that lymphatic, and then we see the fusion, complete overlap, making that nice yellow. So from this, we found many things. We tested all of these markers, but it's very important to note that we were not testing looking at what is lymphatic and what is fusion. Our population was the lymphatic markers, and we were seeing if they have the potential to fuse by having fusion markers. So we found out that 73.74% of those lymphatic cells were exhibiting a fusion marker with SIRT-beta-1, all the way up to 98.92% of those live blood cells that also express STABIN-1. So this is, when looking at this, you might think, oh, well, the STABIN-1 must be way more important than SIRT-beta-1 because it's about 25% more percentage. However, there's two things. One, this was taken at one moment in time. It's transient expression, so whether a cell has fused, will be fusing, or is in the process of fusing, it will probably express different amounts of these fusion markers. So you can't tell at this moment whether STABIN-1 is more important than SIRT-beta-1 or CD36. But also, since it is a 2D image, things can get lost behind other, or it might be on the next slide. So that is why we can't take what is here as complete fact. And then here we see just another representative image. This is showing, it's the same data, just in graphical form. You can see STABIN-1, CD209, both reach over 100% for that double positive cell. And this is really important because if you have a 70% population of something and you have a 90% population of something, it can be assumed that there must be a subpopulation that's expressing at least both of those markers. So there must be some lymphatic cells, some MLECPs, that are expressing multiple markers. So to conclude, the vast majority of these MLECPs are expressing multiple fusogenic regulators. And because of that, we can then infer that these MLECPs have the ability to fuse with the lymphatic endothelial cells, because it does not make sense for the cells to have at least one marker, but potentially more, and not have the ability to fuse, because cells, they do not have markers that they don't use. And then this leads to the question, so you have potential fusion of these MLECPs with the tumor lymphatics, would it then, as we've already said, increase the lymphatic vessel density, therefore increasing metastasis because you're increasing the route of dissemination. If you increase metastasis, you increase the chance of patient death. And this is a fundamentally new concept that is understanding the mechanisms of the tumor spread, and it warrants further investigation. Although what I've just presented might not seem like a big deal, it might seem small. However, if we can target lymphatic vessel density, we can target metastasis, and that could change the way we deal with patient death. I would very much like to acknowledge my wonderful mentor, Dr. Sophia Rand, and of course, my wonderful lab member and grad student in the lab, Swati Athaya, SIU School of Med, including MMICB and Sims Cancer Institute, as well as the amazing Denim Diamonds Gala for funding my research.
Questions for Emma?
So as a breast cancer provider, I know that involvement of lymphatics at the outset predicts for worse outcome for patients. We all see patients with larger tumors that are whose sentinel lymph nodes are negative, without no evidence of lymphatic spread. And those tumors come back to the larger the tumor, the higher the rate of recurrence. Again, not quite as exact as with the lymphatic number of lymph nodes, but it is associated with larger tumors. So it kind of raises the question for me, are these larger tumors producing more of these fusion proteins, and even though there's no evidence of lymphatic spread at the beginning, is that what's ultimately causing these patients to relapse? I can't answer that question, and I don't think you can either. It's just an interesting observation based on your work.
Yeah. Are you suggesting that fusion is making the tumor grow?
Well, I'm just saying that these factors are being produced by the tumor, and as the tumor size is larger, perhaps they're producing more of these that ultimately causes that tumor to get into
some specific markers?
Yes.
Why did you pick those?
Yeah, so we looked at all of... Yes, so it was asked, why did I look at these specific fusion markers? So if I go way back, bear with me. So all of these markers from STATA 1 to CERT Beta 1 have been previously shown in the literature to be involved in osteoclastogenesis, as well as placenta formation, embryogenesis. These are all known fusion markers, which is why we tested them. Although it is important to note, these markers have not actually been looked at with lymphatic markers before. They've been shown time and time again to be involved in fusion, especially in the early 2000s. That was a big part of research. And also potentially involved in cancer in some of these, but never with the lymphatic system. So this is pretty novel research.